Hey, I'm Natalie Grassi. I am a PhD candidate with Murdoch University and Bush Heritage Australia, and I'm here to share my project on wildlife ecology at conservation corridors. First off, I would like to acknowledge that I'm presenting on Whadjuk Noongar country, and my fieldwork will be conducted on Kurang Noongar country. I would like to pay my respects to the elders of these communities, past, present, and emerging, and acknowledge the significant role of Indigenous people in protecting global biodiversity. One key threat to biodiversity is the process of habitat fragmentation. This is the break apart of large contiguous landscapes into small isolated patches, which are separated by a cleared matrix. In a non-fragmented landscape, the individuals within are able to freely move about to seek resources like food, mates, or territory, and the populations within are able to freely intermingle, which enables gene flow and maintains species health. However, in a fragmentation event, these populations become segregated from each other. The agricultural lands that usually bisect these habitats are largely impermeable to our native wildlife, which then presents a significant barrier to movement or dispersal. Those individuals can no longer freely seek resources at alternate patches, and the populations struggle to interconnect. During these events, the size of patches themselves usually decreases, which then reduces the carrying capacity at each. One way this has been combated is through the use of revegetation, which is targeted specifically at improving landscape connectivity. This is usually done through the provision of wildlife corridors. So these are physical linkages between two or more habitat patches. These are either direct linkages or they use a more stepping stone design, which reduce isolation of the landscape by providing satellite habitats between the two large patches. Regardless of their design, the overall aim of these is to improve connectivity and restore the ability of wildlife to freely move through the landscape. And by using revegetation, they also aim to provide new livable habitat to improve those carrying capacities. Corridors can occur at the micro scale. These are usually things like wildlife overpasses, shelter belts, or road reserves. They're usually thin linear connections between two habitat patches. There's also macro corridors. So this is my study site, the Fitzsterling. This is a stepping stone corridor which connects the Stirling Range and the Fitzgerald River National Park. Within that stepping stone corridor, there's also a network of those small linear connections. They aim to just improve the connectivity of some of the patches within. As seems to be a common issue for almost all Australian conservation initiatives, unfortunately the success of corridors is hindered by foxes and cats. So these predators preferentially hunt in open, simple landscapes. These are often associated with cleared matrix or revegetation. These landscapes offer very little resistance to movement, which is ideal for hunting, and any structure that a native animal would usually use for avoidance or escape behaviours often hasn't either grown here or it's been removed entirely. It's because of these animals that the cleared matrix is so difficult for our native wildlife to pass, but they might also be creating ecological traps out of corridors. So corridors essentially funnel the movement of wildlife into these designated revegetated areas. So foxes and cats might simply just be waiting for the native animals to come past. It's been shown that predators influence the habitat selection of prey within one patch. So the edge territory of a patch is essentially a halfway point between edge and core habitat. It's quite simple and open, so predators preferentially hunt here, which then drives native prey into the center of patches. Because of this, they may not actually be able to access the corridors at all. And because most corridors are made of revegetation, they may offer very little structures for escape or avoidance, and therefore might be presenting significant hunting grounds for our introduced predators. There is a difference in corridors between their structural and functional connectivity as well. So structural connectivity is the provision of those physical linkages, such as through the planting of trees. But functional connectivity is the ecological degree to which fauna can actually use those. So for something like a western ringtail possum, their nesting, foraging, and movement is entirely dependent on interconnected canopies of peppermint, jarrah, and she oak trees. If revegetation is not planting those species, or if those revegetated areas have not grown complex enough that their canopies are intertwined, then these areas are not usable to that species. It's not uncommon for a corridor to lack structural connectivity, and it's not necessarily that the wrong things are being planted, it's just that they take so long to grow. So, for example, with black cockatoos, they nest in tree hollows that are at least 1 metre deep, but a tree has to be about 80 to 100 years old before that hollow exists, and so for that time, that habitat is not particularly usable for those species. Using the funds which have been generously supplied by the AWS's University Research Grant, I'm aiming to investigate the synergistic interactions of disturbance, revegetation, and introduced predators, and how this influences the habitat selection of our native fauna. Most of my fieldwork will be used to deploy 128 camera traps within cleared matrix, revegetation, and remnant habitat. This will enable us to compare the fauna compositions at each of these treatments and to investigate which species are able to use cleared matrix for dispersal and which are able to use corridors and those which are being excluded. Using my fieldwork, I'll also be collecting scat and carcass samples of introduced predator for use in a diet analysis. This will enable us to figure out which native species are most at risk. And we'll also be conducting vegetation surveys to enable us to figure out the fine-scale features of habitat complexity, which are enabling our wildlife to persist in these areas. So these fine-scale features are things like leaf litter depth, canopy cover, mid-story cover, and understory density. So thank you very much again for the AWS for your university research grant. Much appreciated. I would also like to thank my supervisors at Murdoch University and Bush Heritage Australia. Please feel free to contact me if you have any questions. And yeah, thank you again.